Hello everyone, welcome to the Dubai's World channel. Today we are going to start a new topic, rabies. Actually, rabies, it is an acute, progressive, encephalitis, and highly fatal disease. Here, this encephalitis means inflammation of a brain. Inflammation in a brain. And it's a zoonotic disease. This rabies word comes from a Latin word that is rabas, which means frenzy. But some books they are also written the Latin words here rabidus, which means mad. So this rabies is also called as a hydrophobia. In a human, basically it's used hydrophobia, which shows or which denotes the fear of water, and the rabid of a dog means saliva of a dog. Basically, this rabies is a disease that is caused by mad dogs which are moving here and there in the streets and a colony. So now we'll see the causal organism. Causal organism. This is caused by Rhabdovirus. Actually, this Rhabdovirus belong to the genus Lysa virus type 1. And this Rhabdovirus is having a lipoprotein enveloped along with the bullet shaped virus, and the size of this virus is approximately 180 into 75 nanometer, and on this viruses there is a matrix protein layer, means it's covered by matrix protein layer. When we'll see the genome of this virus, genome is unsegmented, linear and contain single strand RNA with the negative sense. These viruses also have knob-like spikes on the body surfaces. So now we'll see the pathogenesis. pathogenesis here, this how this virus infect the a host after biting of a mad dogs. So after biting a mad dogs, so before going to pathogenesis, we'll see who are the host and reservoir for rabies virus. Basically, the mammals are the natural host for this viruses. Mammals, first mammals, and seconds all the warm-blooded vertebrates. And mostly the reservoir who contain this type of viruses are cat, dog, bat, etc. So these are the hosts which consist of this rabies viruses. Now we'll start with the pathogenesis. Basically the incubation period for this rabies viruses is 20 to 90 days. So how this viruses transmit to the human body or a host and infect the cells. So first the virus inoculated by bite, means when the dogs bite to the human or a host, the virus inoculated by bite. After this, virus replicated in the muscles at the site of bite, means where the dog has bite already, there only the multiplication and the virus will multiply, the site of bite. After this, virus infect nerves in a peripheral nervous system, which moves by retrograde axonal transport. Then virus replicate in a dorsal root ganglion and travel up to a spinal cord to brain. After this infector brain is going to infected and the CNS will is disseminated. After this, once the brain is going to infected, the virus travel from brain via nervous to other tissue such as kidney, salivary gland, adrenal gland. Once they travel to this other tissue, it invades all this tissue, all these organs like a kidney, salivary gland, and adrenal gland. Also, cardiac muscles, and also invade the skeletal. So this virus inoculated by bite. Once the dog will bite that host. At the place the virus will replicate. After that, after the replication, the virus infects nerves in the peripheral nerves system and moves up by retrograde axonal transport. After this, it will replicate in the dorsal root ganglion and travel up the spinal cord to the brain, and there the brain is going to infect it. After the brain, the virus travel from brain via nerves to other tissue and invade all this tissue. Then we'll see the lab diagnosis of a rabies disease. Here first we'll do the saliva test, then lumbar puncture test. In that, what they are going to do, they are going to take a specimen from with the needle. They are going to take a CSF, a cerebrospinal fluid, or a saliva, and they will do the biopsy, where they will come to know about this rabies virus antigens are present or not. And third, they will do the immunofluorescent test, immunofluorescent technique. In that, the antigen, the rabies antigen are going to stain, and will absorb under the immunofluorescent microscope, means a fluorescence microscope, where the antigen is going to be stained, and there it will bind to the antibody, and there they will check the viruses, this rabies viruses. One more technique is there. We'll do the PCR, polymerase chain reaction will use, and there they will get the information about the antigen of the viruses, rabies viruses. Now we'll see the treatment. Actually, there is no any established treatment for this, but for relaxation purpose, there should be this patient should be kept in a quiet room. Patient should be kept in a quiet room, as bright light, noise, cold, draughts, and peptispasm that will cause a convulsion to the patient. And at the time, sedatives should be given to relieve the anxiety, and the proper hydration should be given. And the intensive respiratory and cardiac support should be given, Now we'll see the prevention. Here, once the dog will bite that person, that this prevention consists of there's a two way to prevent the host from this viruses. This is a pre-exposure and post-exposure. Post-exposure prophylaxis, pre-exposure and the post-exposure prophylaxis. Here in this pre-exposure, what they will do, they will clean the health. A person who don't have any wound or any cut, that person only should treat any human being that already got bited by a dog. So person should be wear the gloves while during a treatment. A person should wear a gloves and the mask. Along with that, they should clean the portion where the dog already has a bite, and then and then it should be cleaned with the soap and a water or a solution of a quaternary ammonium compound, because this material destroys the rabies virus effectively. After that, half of 20 in this pre-exposure. In post-exposure, after that, half of a 20 UI/kg body weight dose of a human rabies immunoglobulin, or a 40 IU/kg, it's a unit, IU per kg is a unit, should be given either 20 IU/kg or a 40 IU/kg antibody or immunoglobulin should be given to the patient, and after that should be administered, and that should be intramuscularly, not intradermally, intramuscularly in a gluteal region. This should be given in a gluteal region, or remaining of the both doses should be infiltrated around the this bite site, means where the already dog has bitten, and then 1 ml of potent cell culture vaccine should be given intramuscularly on the day of 0, 3, 7, 14, 30 and 1990 in the deleted region. Rabies antibody test may be done on the day of 30 to ensure that whether the patient has developed a satisfactory response or not. In the pre-exposure prophylaxis, the one who is the relative or one who is taking care of the person, infected person, should also check whether that dog now the dog is cured or not, or a dog is in a stable form or not, or it is died. So at the time of this pre-exposure prophylaxis, that person who's curing the infected person, that also should also check about the dog. If the dog was killed, or if dog is died, it's a negative for a rabies by a laboratory diagnosis. So it should be watched. It should be seen that whether the dog is become healthy or any different changes, or it's killed. So we have to that person should check the behavior of a dog also. So now once again I will say, the prevention consists of a pre-exposure prophylaxis and post-exposure prophylaxis. In pre-exposure prophylaxis, a person should be one who cured the infected person should clean the bite site of where the dog has bite. So clean, and then with the soap or water or a quaternary ammonium. I will write soap and water or a solution of a quaternary ammonium, because it's effective. This compound destroys the rabies virus effectively. In the post-exposure, these 2 doses of vaccine should be given in a gluteal region, or another vaccine, that is 1 ml potent cell culture vaccine, is given on a first day, third day, 7th, 14th, 30 and 19. On the 30 day they can do the rabies antibody test to check is that the vaccines are given to that person is satisfactory or not. Rabies antibody test. This is for rabies virus. Thank you.